Hi, my name is James Latimer. I'm the lab manager of this facility, which is the wet lab for the Fenner School of Environment and Society. Here we do a lot of wet chemistry, so we do a lot of nutrient analyses and say rainwater and environmental samples. We deal with water and soil and rocks and plants and animal samples. So one of our instruments here is a microplate reader. So here we can analyse things like nitrate concentration or ammonium concentration. Other wet chemistry we do here, we do acid digestions and titrations and standard wet chemistry procedures. We have fume cupboards, both scrubbed and regular, centrifuges, fridges, standard equipment. Here we have a microwave digester. So this is an instrument for liquefying samples. So we can liquefy rocks and plastics and soils, which makes further analyses much easier on different instruments that we own. Here we have our undergraduate teaching lab. So we have class sets of microscopes and soil samples and different demonstration tools that we can use to teach in undergraduate subjects. This is our Dendrochronology and entomology lab. So here we have, we do a lot of tree ring dating. So we have some beautiful pieces of wood that have been precisely cut and polished so you can count the rings well. And we do a lot of insect experiments here as well. Here we have a olfactometer, which is a device for exposing insects to different aromas and then recording their behaviour depending on what you expose them to. We have two nutrient analyzers. So we have a flow analyzer that measures things like nitrate and phosphorus. And here we have a carbon combustion analyzer. So it measures carbon and nitrogen. In here we have equipment like these biological containment chambers. So they filter the air in and out so you can work with hazardous materials and contain that material. Here we have another type of hazardous containment chamber. And then some workstations here. So there's a lot of standard microbiology work that happens here. So pipetting and microplates and various incubations and centrifuging and standard microbiology and genetics work.